Yeah. So good morning, everybody. I am going to talk about improving the duration of immunity of conventional FMD vaccine. So before going to talk about the vaccine, so we should know what are the problem and what are the issues we are having with the conventional vaccine and what we are trying to solve in this particular study. So as you know, and we are discussing since last 3 days, this is a killed inactivated vaccine and adjuvanted with mostly with oil. And in the end, as I said in my title, it is mainly I'm talking about endemic setting, and biannual vaccination is usually practiced, and with 3 PD50 vaccines. And one of the major challenges is short-lived immunity, which provide maximum 3 to 4 months, and that allows a big window for at least 2 months before the second vaccination. So this is the main aim to address how we can cover these 2 months or 3 months window before the vaccination, so that we will not get, our animal will be not susceptible again to bring back the FMD viruses. So with this aim, another secondary aim is, as you know, that FMD vaccine provides mainly Th2 response, that means mainly antibody mediated, as we see yesterday from different talks, and it doesn't give much Th1 response, and by using different adjuvants, we are also trying to increase the Th1 response, that is mainly cell mediated response along with humoral responses. And I will bring you back to our work in 2012. So in the first left-hand side of our panel, I am taking you to two vaccines which we tested at Purbright. The black bars shows the BNT for the A22 vaccine, whereas the white bar shows the BNT for the SARS-2 vaccines. And this is the usual European pharmacopoeia potency with different doses. I'm not going to describe. You can see both the humoral antibody are in both vaccines are almost similar, and whereas 4 animals in SARS-2 vaccine, including the full dose group, were not clinically protected. So doing more work on that to check the Th1 response, so we did the interferon gamma response. As you can see, in all the animals in A22 vaccine were protected, and also they were having very high interferon gamma response. That is the black line. And same time, if you see the white line for the SARS-2 cell-mediated response, that is interferon gamma, which is very flat with very low or very little interferon gamma response. Then we tried to correlate this interferon gamma as well as VNT with the clinical protection. You can see in the right-hand panel in the upper, so it correlates well both with interferon gamma as well as neutralizing antibody. Then we were interested to know, so whether this interferon gamma is mediated or produced by which cells, like CD4 or CD8. In the first panel, in the left-hand side down, so you can see when we depleted CD4 cells from the total PBMC, and this interferon gamma line has drastically reduced. And on the right-hand side panel in the down, so you can see when we have taken out CD8 cell depleted, and it has been reduced, but not much. So as you know from our vaccine, that it gives very little CD8 response and more CD4 response. So above all, the cell-mediated response is low. So the main aim was to increase the cell-mediated response and along with the duration of immunity. So we have chosen a variety of adjuvants. So here, 8 adjuvants, and they are really used in human medicine. So they are very efficient. So some of them were TLRs. I will show you different adjuvants later on. There is no hidden A and B or adjuvant 1 or 2. So I will show you exactly what we got. So when we take this new generation adjuvant, this activate the innate immune system, as you know, via the binding, ligand binding to PRR, and enhance the antiviral environment, and also enhance the adaptive immune response, which I am talking, CMI as well as humoral antibody, and finally improve the memory response. So that's why what we did, we took 80 adjuvants, including TLRs, and then we did a pilot study at the beginning at Indian Immunologicals few years before. And we have presented this in a few meetings. And if you see here, what we did to test this potency of the adjuvant, how potent they are, we have not taken full dose of antigen. So we have taken, this is the most important thing, we have taken the suboptimal dose of antigen. So basically, people take in endemic country 10 microgram of O antigen. So we have reduced here to half, 5 microgram of antigen. And then we have taken different, the first one is Avisco, which is a ISCON matrix. And the second one, we have taken CPG, which is TLR9. And we have taken the ISA 70, which is similar to ISA 206, our oil. And then we have taken poly-IC, which is a TLR3. And we have taken imiquimod, which is a TLR7. And then we have taken MPLA, which is a TLR4. And also we have taken liposomes and the usual oil, ISA 206 as a control with 5 microgram antigen, and also with the full dose 10 microgram of antigen. And if you see the percentage of protection, in Avisco we got 100%, and in poly-IC we got 75%, and in imiquimod we got 75, and in MPLA 75. And in oil, 206 with 5 microgram we got only 50%, whereas with full dose we also got 100%. So now we narrowed down to these 4 adjuvants. I have put it in red star. So you can see the Avisco and this poly-IC TLR3, imiquimod, this is the TLR7, and MPLA TLR4. So we took this one to Purbright to do another serotype experiment in a detailed manner. So this is now we switch to type A with A22 antigen. And as you saw here, again, as you know, so for A serotype, people or industry take a little bit less antigen. Like I said, 8 to 10 microgram antigen for O, but A is stable, so better stable than the O. That's why people take like a 6 to 8 microgram. So I have taken here only 2 microgram, because my aim is to know which adjuvant will help. So otherwise, we cannot differentiate between the adjuvant. So as you can see here, I'm showing the percentage of protection again with the larger number of animals, now 5 in each group. So you can see there's Avisco given as last time 100% protection. And then poly-IC gave also 100% protection. Now this imiquimod and MPLA, they gave very less protection, like 80% and 40% protection for A serotype. So what we did, this is the plan of the experiment, what I showed in a final slide before this. And we have vaccinated, and then we have challenged in 21 days vaccination. Then, sorry, it was actually not 21 days because due to some problem, so we could not challenge it to 21 days. We challenged on 30 days. And then we called the animals on 30 days, and we collected samples daily, including the daily rectal samples, nasal and saliva swabs, clotted blood, and heparinized blood. And if you will see in left-hand side panel, I am showing the genome copy excreted in the nasal swabs. And the right-hand side, I am showing the replication of the virus through NSP antibodies. So you can see in the all groups, still there is subclinical replications. So not, it is similar to our existing vaccine. So then we have done the FACS analysis, and you can see in the bottom panel we have increased the both CD4 and CD8 response in comparison to ISA 206, which is in the second and blue in color, which was very less. And here we adding the poly-IC, and we have added in the poly-IC group, it is a little bit higher than other groups. So then coming to the antibody response, the humoral antibody response, both poly-IC and Abisco are having very significantly higher antibody than the oil group, ISA 206. So then my first summary is we achieved an increased potency as indicated by lack of clinical symptom in poly-IC and Abisco group. And although we could not prevent the subclinical infection, and significantly elevated a neutralizing antibody in Abisco as well as a poly-IC group, and some indication of interferon gamma upregulation by CD4 and CD8 cells we have seen. So now what we did after we selected these two, so we talked to the company ISCON Matrix or Abisco, and we were not able to get that one anymore. That company has been taken by somebody else, and they were not happy for the testing in animal. They said, okay, we are working only for human beings, so we didn't get that vaccine, that adjuvant. So we continued with the poly-IC, which we would have been chosen because poly-IC gave both cell-mediated as well as humoral. So whereas Abisco gave humoral fully and very little cell-mediated, so that was our chosen adjuvant. So we have taken forward this one now, again went back to Indian Immunologicals, and we have taken a larger group of animal because we cannot do this work in provide keeping the animals for 7 months. So the original plan was to keep 6 months and just to challenge after the 6 months, but we are not lucky to challenge it due to some regulatory procedures at Indian Immunologicals from the government, their government. So what we did, we have vaccinated 11 animals with the poly-IC and oil. So all the talk I'm giving, always oil is there, and we are adding the poly-IC. Or any adjuvant. We have not taken out our oil because we have seen oil is giving very good humoral antibody. So that's why we are adding every time. So same as here in the longer duration immunity. So this is the first group is with the ISA, with the poly-IC, and second group is our usual vaccine with oil. And we have taken now the full dose of antigen, that is 10 microgram of antigen. And what we have done, so we have vaccinated and kept the animal up to 7.5 months and collected samples, serum, PBMC, and antigen specific stimulated whole blood plasma and transported to provide to do all the assay. Here I am showing all the virus neutralization tests for individual animal. I would like to show how the individual animal works. So for each month from 0 day to 7.5 months. As you can see, by 120 days, which I cannot have a pointer, you can see here 120 days, all the red ones are our oil, and the blue ones are oil and poly-IC. So there is a significant difference. Simon has done a mess, and you can see all the group of poly-IC is significantly different than the oil. And then doing the whole body interferon gamma, and I would like to show again the individual animals, and from every month we have done. You can see the red one here, the conventional with oil, and the second one is with oil and poly-IC. And you can see also again Simon has done the analysis, and this is significantly different in each time point, leaving the 30 days and 150 days. So they are always highly significant in poly-IC than the conventional vaccine. Then we have done also the FACS analysis, and the first one is for the group 1 conventional vaccine, and the second one is for the poly-IC group. And what we have done here, this is the population of CD4 and CD8 internally expressing interferon gamma out of the total population of PBMC, not of the total population of CD4, CD8. So as you can see, poly-IC group again is having higher response than the OL group. So my final summary is significantly higher VN titer in cattle vaccinated with poly-IC is seen compared with conventional adjuvant. And by 4 months, many of the conventional vaccinated animal lost the protective level of antibody titer. If you know, so in South American people, they showed it is a log 1.65 protection value. So you can count from them. Then interferon gamma level significantly higher in cattle when vaccinated with poly-IC when compared with the conventional adjuvant. Then indication of interferon gamma upregulation by CD4 and CD8 cells were observed in poly-IC group than the conventional group in FACS analysis. So my question at the beginning, duration of immunity can be enhanced up to 6 months minimum. So post-vaccination that covers the window of susceptibility before the second vaccination, so that we don't need a booster dose or which is proposed usually, or there it should not bring the outbreaks to the population in the endemic country. With this, I would like to acknowledge those who have done work with different serotypes at Purbright and also at Indian Immunologicals. So they are here. I'm not going to say much about them. And Simon, I would like to thank him for all the statistical analysis. And I would also like to thank David and Geraldine, who are the collaborator in the project. And it will be nice to acknowledge Indian Immunologicals for their help, and Dr. Srinivasan particularly helping at the time, agreeing to do these experiments there, and later on taking with the other managers from Indian Immunologicals. And I would like to thank BBSRC and also DFID UK for providing 2 BBSRC projects to carry out all these animal experiments. Thank you for your attention.
Thank you.